The overall goal of this assay is to observe individual membrane fusion events driven by neuronal and exocytotic SNARE proteins to understand fundamental mechanisms of neurotransmitter and hormone release.
This method can help answer key questions in the membrane fusion field, such as how cargo release occurs through a dynamic fusion pore and the fate of the pore after fusion.
The main advantage of this technique is that distinct stages of vesicle docking, fusion pore opening, and cargo release kinetics can be observed for individual events within biochemically defined environments.
Though this method can provide insight into exocytosis, it can also be applied to other membrane fusion reactions, such as the fusion of envelope viruses with target cell membranes during infection.
Generally, individuals new to this method will struggle because many different steps need to be mastered, from microfabrication to protein purification and imaging.
To begin, prepare a silicon master mold using standard photolithography techniques and the template shown here. Next, add 100 milliliters of PDMS base and 10 milliliters of the curing agent into a disposable plastic cup and stir the mixture well. Then, place the cup into a vacuum desiccator. Apply a vacuum, but watch it closely to prevent the mixture from overflowing. Once degassed, pour a large drop of the degassed PDMS mixture into a glass petri dish and press the mold onto the PDMS with the template side facing up to avoid trapping bubbles beneath the wafer. Now, pour degassed PDMS onto the template until it is covered by 5 to 8 millimeters of the polymer. If an air bubble occurs, gently remove it with a pipette tip.